Well, hello there, everybody. We are now back to part 4 of our metabolism lecture. Here we're going to talk about the primary metabolic pathway, which consists of glycolysis, something known as the transition step, which includes the breakdown of pyruvate, the citric acid cycle, and a process known as oxidative phosphorylation. So glycolysis occurs in 2 stages. The first is an energy investment stage where we actually are going to have to input a little bit of energy. And then the second stage of glycolysis is the energy release stage where we actually are going to profit a little bit of energy from glycolysis. And glycolysis is a metabolic process that is able to occur with or without oxygen. It happens in the cytoplasm of eukaryotic cells and in prokaryotic cells. The steps in glycolysis are common. They are what we call conserved processes. They are conserved processes across most species. So in the world, we process our glucose the same way that bacteria do, and dogs and cats and other animals and even plants. There are 10 steps. Those 3 phases within the steps. The cleavage phase is not always considered a phase, but we'll go with it for this book. So there's the energy investment phase where we have to add a little bit of ATP to get things going. Then the cleavage phase is when the glucose molecule is split in half. And then energy liberation is when ATP is actually produced. So in the energy investment phase, this consists of steps 1 through 3. 2 ATP will be hydrolyzed, or their phosphate groups will be broken out to create a molecule called fructose 1,6-bisphosphate. I don't get into naming and memorizing all of the chemical names and enzyme names and such. I do want you to know maybe the first enzyme in this process, the hexokinase. And you should be familiar with how the organic molecules are named, but you do not have to memorize all of them. So the first molecule we're looking at here is fructose 1,6-bisphosphate, and this is because ATP will lose one of its phosphate groups here, and it will be added to a linear molecule of glucose, and this rearrangement in glucose will cause it to become a fructose molecule, and we'll have actually 2 ATPs are used, so we get an ATP adds a phosphate group to carbon 1, the other one adds a phosphate group to carbon 6, and this is now a 2-phosphate fructose molecule. So that's why it's named the way that it is. In the cleavage step, this consists of steps 4 and 5. Now, glucose is a 6-carbon molecule, so when we cut it in half, we're going to get 2 3-carbon molecules. In the energy liberation stage, this occurs in a couple of steps. It is steps 6 through 10. And we get 2 molecules broken down, those 2 3-carbon molecules. They're called G3P, or glyceraldehyde 3-phosphate. You'll see G3P is what they call them for short, and you're going to see this molecule quite a bit. Plants make it, our cells make it, animal cells, bacteria cells even make it. And these molecules are going to get broken down into 2 molecules known as pyruvate or pyruvic acid. During that process, pyruvate is our end process of is the end product of glycolysis. So at the end of glycolysis, let's put GLY here, we get 2 ATP. Those are going to be made using substrate level phosphorylation. We're going to get 2 pyruvates. These are the end product of glycolysis, and they are going to move through another step in metabolism. And then we're finally going to get 2 NADH. If you remember from the previous lecture, the NADH, these are energy intermediates that are going to carry electrons to the electron transport chain. So here is glycolysis. Here you can see, this is a diagram from your book. Here's glucose in the beginning. In step 1, we add a phosphate from ATP. In step 3, we add another phosphate from ATP, and this is that fructose 1. This is 1 over here, 6 bisphosphate. This molecule will be split in half And steps 4 and 5 are going to occur down here as well. So here, this molecule gets split in half, and here I end up with my two G3P molecules. Each one of these will lose an electron, so I get an NADH. Each one of them will carry out substrate level phosphorylation, and they will do this twice for a total of 4 ATP. Now I know I made 4 before, but remember we had to input 2, and 4 minus 2 equals 2. Here is where we have that first stage of glycolysis before the cleavage phase, and this is just going through each of the individual steps. These are the intermediates, okay? So sometimes they're called the precursor metabolites, and so those molecules are going to match up with these enzymes right here. Here's all the enzymes. You do not need to know the names of all these, but these enzymes carry out specific functions, and that's why the molecule must meet up with the enzyme prior to moving through the pathway. You should be able to read a metabolic pathway and answer questions regarding it. This is the last phase, so you can see here instead of ATP going in, now I have ATP coming out. That's what this curved arrow means here. And at the end of this, I end up with 2-pyruvate. And of course, I get 2 ATP because remember my glucose molecule got split in half. So this always happens twice. And I have 4 ATP gross, but I have 2 ATP net because I had to input 2 ATP at the beginning of the process. In stage 2, we're going to break down pyruvate into what's known as acetyl-CoA. This is referred to as an acetyl group. So in eukaryotic organisms, our pyruvate, once it's made in the cytoplasm of the cell, is going to get transported over into the mitochondria. The rest of our primary metabolism is going to take place inside the mitochondria. In the mitochondria, pyruvate will be broken down by an enzyme called pyruvate dehydrogenase. It's going to cause the release of a CO2 molecule, so we lose some CO2. And whenever you see a CO2 being lost in any of these pathways, this is usually going to result in the production of an energy intermediate. I'm just going to put EI for energy intermediate. In this case, our energy intermediate is going to be NADH. There's an enzyme also known as acetyl-CoA, and so this new acetyl group is going to get attached to enzyme CoA, and this gives us the molecule acetyl-CoA. Acetyl-CoA is going to enter into the Krebs cycle, and our products here are going to be 2CO2 and 2NADH. NADH is going to leave and go to the electron transport chain. So here we have our pyruvate molecule, right? 3-carbon pyruvate molecule moves into the inner matrix of the mitochondria, meets up with pyruvate dehydrogenase, meets up with CoA. This is an acetyl group here, and it becomes the molecule acetyl-CoA. Here is our products, which is CO2 and NADH. The citric acid cycle, also known as the Krebs cycle, and I believe your textbook calls it the tricarboxylic acid cycle, or TCA cycle. In this case, instead of being a linear process, this is a cyclic metabolic pathway. So we don't really have any true end products. The end product becomes the beginning, one of the beginning reactants in the cycle itself. So this is a circular or cyclic metabolic process. During this process, some of our intermediate molecules, our precursor metabolites, may leave, others may enter. And so this causes a very complex reassortment of organic molecules that get regenerated. Acetyl will first be removed from that acetyl-CoA. So what was glucose originally in glycolysis converted to pyruvate, then converted to acetyl-CoA, now enters into the Krebs cycle and is attached to a molecule called oxaloacetate. You do need to know this molecule. Oxaloacetate will bind with acetyl-CoA and form citrate, also known as citric acid. This is why it's called the citric acid cycle. Through a series of long and complex steps, ultimately the products we will get from the Krebs cycle are 2 carbon dioxides, 1 ATP, 3 NADH, 1 FADH2. Remember, this is another energy intermediate. All of these processes are going to be times 2. And this is because, remember, pyruvate or glycolysis was split into 2 pyruvate molecules. So 2 pyruvates equals 2 acetyl-CoAs. 2 acetyl-CoAs entering into the citric acid cycle will cause this to be doubled. So for every glucose molecule that enters into primary metabolism, we actually end up with 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. And you can see from here that the primary function of our Krebs cycle here is not production of ATP, but production of NADH and FADH2. At the end of our Krebs cycle, the molecule that was rearranged, the acetyl-CoA that entered and got rearranged, will ultimately end up rearranged back into an oxaloacetate. So that particular molecule will be regenerated to begin the cycle again with the next incoming acetyl-CoA. So here's our citric acid cycle. It's taking place in the inner matrix of the mitochondria. So it's inside the mitochondria here. And every time an acetyl-CoA enters, we get an NADH here and a CO2. We get NADH and a CO2. We get an ATP molecule. It actually comes out of the citric acid cycle as GTP, but it ultimately is converted into ATP. In step 6 here, we get another energy intermediate, FADH2. And then finally, at the very end, we end up with another NADH. And then this right here, this 4-carbon molecule right here is oxaloacetate, which will combine with the incoming acetyl-CoA to give us citric acid. So that's a single turn. Remember, glucose is split into 2 pyruvates. So glucose becomes pyruvate. And each pyruvate becomes an acetyl-CoA. And then each of these are going to enter into the Krebs cycle. So this becomes 2, 2, 2, 2, 2, and 2, and 2. So that becomes, you can see 2, right? So 2, 4, 6 NADH, 2 FADH2. Oh, sorry. Oh, I lost my. And then we have 2 ATP. Here's another look at the Krebs cycle with a little more detail, showing you what's happening in each of these processes. Here's our acetyl-CoA, citrate synthetase. That's the oxaloacetate right here. So this enzyme is combining these 2 molecules together. Oxaloacetate is not an enzyme. It's a molecule. It is a precursor metabolite, just like the malate and citric acid and such are. So this molecule is combined with this molecule. And we have these groups right here. You can see these carboxyl groups. And you see this function right here, and here they are. And they get attached. So this guy gets attached right up here. Then we end up moving it. It gets changed around. And the idea here is to follow the carbons. So when it's still a 6-carbon molecule, but when we start rearranging this molecule, look, it goes to a double bond here. This NAD plus receives that electron, becomes an NADH. A carbon is lost because here it's a 6-carbon molecule. Here it's a 5-carbon molecule. I lost a carbon and 2 oxygens, that's CO2. Same thing happens over here, right? So I've gone from a 5-carbon molecule to a 4-carbon molecule, and I get another CO2 molecule. So whenever you have a CO2 molecule, it's because you're ripping a carbon off of the original molecule in the pathway. And the body's got to do something with it, so it converts it into CO2, and we exhale it. So the apple or banana that you ate this morning for breakfast is now the carbon dioxide being released when you exhale. And that is why your blood will bring oxygen to your cells and remove CO2 from your cells. That CO2 waste is gaseous waste that's coming from your metabolism. Now, stage 4 is what's known as oxidative phosphorylation, and this is where we make the most ATP. Oxidative phosphorylation is where electrons that were removed from glucose and added to NADH and FADH2 now come to the electron transport chain in order to create electrochemical gradients, and that's going to cause the production of ATP. Now, oxidative phosphorylation most commonly will use oxygen because oxygen has the greatest electronegativity, and it's going to pull electrons through the electron transport chain with much greater power and strength than, say, a carbon molecule or a sulfur atom. So oxygen is typically what's used at the end of the electron transport chain. In anaerobic conditions, however, a different atom is going to be used. The process of oxidation occurs because the electrons are being shifted around. So NAD plus goes out into the cell, it grabs an electron, gets reduced, and becomes NADH. NADH, in turn, goes to the electron transport chain, gets oxidized, and then becomes NAD plus. The NAD plus will then go back out into the cell, find another electron, become NADH, go into the electron transport chain, drop off that electron, and go back out into the cell and keep doing that process over and over again. Phosphorylation in this case is where ADP will have what we call a free phosphate added to it, and that will give us ATP. This process is known as phosphorylation when you add a phosphate to a molecule, and this is going to occur within an enzyme called ATP synthase for synthesis of ATP. Now, the electron transport chain is a group of proteins. There are 4 of them. There's really 5 or 6 of them, but there are 3 that are extremely important because they are what we refer to as proton pumps. So we have this large group of protein complexes. It will include some attached organic molecules, and they are found all throughout the inner mitochondrial membrane, that inner membrane inside the mitochondria, which is why that membrane is invaginated or bent up, right? So the outer membrane kind of looks like that. Mitochondria looks like a pill, but the inner membrane has all this crazy folding that goes on. It's all wrinkled up like that, and that is to increase surface area. The more surface area there is inside of the mitochondria, the greater the number of protein complexes or electron transport chains available for ATP production. In the electron transport chain, redox reactions are constantly occurring. So the electron transport chain can accept and donate electrons, and it does this in a very specific order, kind of like a set of dominoes. And as these electrons move across the membrane through these protein complexes, it's going to create an electrochemical gradient. That electrochemical gradient is known as the proton motive force. And the proton motive force is going to provide the energy for ATP synthase to actually phosphorylate ADP. So we have this excess of positive charge, and that's going to be going on out here, and then we have charge in here, and we end up with this electrochemical gradient. So here's the mitochondria right here, right? And so you can see it's all enfolded and all that here. And this is a close-up of the electron transport chain. So the very first thing that happens is this looks very confusing and really there's a lot going on here, so hopefully I can ease your mind a little bit and walk you through it. So when NADH picks up an electron from either glycolysis or the Krebs cycle, it's going to bring it over here to the electron transport chain, and it's going to drop off or release that electron here with this NADH dehydrogenase, it's called. So we call it complex 1. So it drops off its electron at complex 1, becomes NAD+, and goes back out into the cell to repeat that process again. NADH's job is now done. It is NAD+, and the electron is released into this enzyme here, NADH dehydrogenase, or complex 1. Now, electrons are super, super high energy. So think of them as big hot potatoes. This potato right now is really super hot, and this enzyme cannot hang on to it for very long. So this little guy right here, called ubiquinone, is going to grab this electron. And so this guy's going to swing over here, pick up the electron, and then swing back and take this electron to complex 3. This complex down here, complex 2, will get skipped. Now, complex 1 and complex 3 both have absorbed some of the energy that this electron is giving off. And when they absorb that energy, they use it to pump protons across the membrane. This is a secondary transport, and it's active transport. Electrons are providing energy for the proton pumps to function, and that's what these complexes in the electron transport chain are. They are proton pumps. They're going to move protons from one side of the membrane to the other, and ultimately we're going to build up a really large concentration gradient of all these hydrogen ions, just put plus signs for now, on this side of the membrane. So I build up this strong hydrogen gradient, this ion gradient. Now, this complex 3 can't hang on to that hot potato for very long either. And this molecule here, cytochrome C, is going to grab it and transport it over here to cytochrome C oxidase, or complex 4. Complex 4 will also pump some protons across, so I end up with 3 proton pumps. I have 3 proton pumps in the electron transport chain. They're going to keep pumping hydrogen ions across. Eventually, this electron, however, is going to get accepted by an oxygen atom. So oxygen is down here. We breathe in oxygen. Our red blood cells deliver lots and lots of oxygen to our cells for an extended period of time. Our cells need a lot of oxygen. And the reason for that is for all of these protons that are getting pumped across, electrons need to be accepted by oxygen. So oxygen is what we call the terminal electron acceptor. And this is in aerobic cellular respiration. Acceptor, right? And this is in aerobic. Now, if this was anaerobic, instead of oxygen, it would be just something else, carbon, sulfur, nitrogen. Those atoms are all oftentimes used in anaerobic processes. So we have this cytochrome C oxidase that is also pumping hydrogen ions. So I've created even greater gradient across my membrane here. And now I have this really strong proton gradient on one side of my membrane and a very low proton gradient on the other. So what's going to eventually happen is, what these blue lines are showing you, is that these hydrogen atoms are going to diffuse back across the membrane. And ATP synthase is what allows this to happen. Cell membranes are phospholipid bilayers. They are not permeable to protons or electrons. Those two subatomic particles have far too strong of a charge to be able to cross a phospholipid bilayer on their own. If they are going to diffuse across that membrane, they have to have some sort of channel in which to do that. Protons are charged particles just like electrons are, so they have energy. And all this energy that's built up on one side of the membrane is now going to move through an enzyme called ATP synthase, who is going to use that energy, harness it, to create ATP. Now, in the Krebs cycle, in addition to NADH, a second energy intermediate was produced, and that's called FADH2. FADH2 will produce electrons as well. However, it drops its electrons off at subunit 2. And subunit 2 immediately sends the electron to subunit 3, or complex 3. Because of this, FADH2 skips complex 1, which is a proton pump. So FADH2 only runs 2 proton pumps. Whereas, you get the idea. My writing tool is giving me a hard time here. NADH, however, runs 3. So if NADH runs 1 more proton pump than FADH2 does, then my NADH is going to make more ATP. It provides more energy because it provides more protons. So the trick here is to see the connection between all of them. See the connection that's occurring between all the different complexes in the, my pen does not want to cooperate with me here, that occurs between all of the different, let's erase some of this, it's getting a little hairy here. There we go. So we want to make sure that we are following the electrons and that we are that we're creating this gradient over here. So in aerobic, that's what I circled right here, oxygen is used. In anaerobic, anything but oxygen is going to be used. Okay. When this is occurring, the movement of NADH to oxygen is what we call a negative free energy change. So it's spontaneous. It's going to cause our reactions to move in a forward direction, and it's highly exergonic. Because it's exergonic, some of that energy can be used to pump those protons across that inner membrane, creating our proton electrochemical gradient. This is just a graph from your textbook that kind of shows you this free energy exchange. Here we have complex 1, and right, so here's complex 1, complex 2, and complex 4. Right? Or I'm sorry, complex 3 and complex 4. And this is showing from, notice, from NADH. Complex 2 is not being shown. Okay? Complex 2 is showing, would show up somewhere over here because it interacts directly with ubiquinone, and this is your FADH2. And you can see this falls lower on the free energy exchange, so it's going, FADH2 is going to produce less energy than NADH. Now, ATP synthase is an enzyme, and it takes energy from the flow of those protons as they want to diffuse. Diffusion is a natural process, so no energy input is required for that. The energy input was required to build the concentration gradient. Once the concentration gradient is built, we just maintain the concentration gradient, and diffusion is going to naturally occur. So those hydrogen ions, those protons, they want to move back across the membrane to where there's a low concentration. And in this case, they're going to release energy in that process. So really what's going on is these protons all get kind of shoved up on one side of the membrane, and then ATP synthase is just the gate. It's the only way for those protons to move back across the membrane. They can't diffuse freely because it's a phospholipid bilayer, so they have to diffuse through ATP synthase. And so ATP synthase really kind of harnesses all those protons as they're moving through, and those protons, they're charged. And if they have charge, this means they have energy. And just simply the movement of those charged molecules through this enzyme is going to give it enough energy for it to start synthesizing ATP. So ATP and free phosphates bind with very low affinity, meaning they're, the bind is there. The free phosphate group binds to ADP, but ADP already has a bunch of negative charges on it. And the free phosphate group is also negatively charged. So when we have to charge these 3 phosphate groups onto each other, it really takes a lot of energy to do that. But that's also why it's such a reactive molecule and works so well to release energy for cellular processes. Here's what ATP synthase looks like. So this side over here, they have this upside down to me, but this side over here has a really high proton gradient. That's what I'm drawing these little plus signs for. And these protons are going to move, they're going to diffuse through this little tube-like structure of ATP synthase. As they do that, it's going to provide energy for this section right here to literally rotate. And ATP production takes place right here. And this arm is going to come off and then go back on, and then come off and go back on. And this guy here will rotate. Every time so many protons move through ATP synthase, this rotary section of that enzyme is going to literally change shape. It's going to rotate, and each rotation results in ATP production. So I'm going to see if this will play. Yeah, my videos are not playing right now, so we will. I'll try to put this up in D2L for you. Now, other organic molecules can be broken down. Sugar is not the only thing that we break down, right? So we have carbohydrates, and there are lots of different kinds of carbohydrates. Ultimately, all of them are going to be converted into some kind of glucose for glycolysis. So sugars will always enter into glycolysis. Proteins, when if we don't eat any carbohydrates, then the first thing we're going to break down actually are fats. That's the idea behind dieting, right? Cut back your sugar, and your body will start depending on your stored energy in the form of fat. Now, fats are basically, if you remember from Unit 2, they're a glycerol molecule with a long chain, with a long fatty acid chain. Fatty acid chains are just carbons, right? Big, long hydrocarbon chains, carbons and hydrogens, right? They have these big, long chains. Now, when fats get broken down, the glycerol molecule gets broken off of the fat molecule, whether it's a diglyceride or triglyceride, and it will take that glycerol molecule, and that glycerol molecule enters about 3 or 4 steps, right? So about step 4, this is G3P. It will be converted into a G3P molecule and enter into glycolysis just after the cleavage stage. The fatty acid chain will skip glycolysis altogether, be broken down into a whole bunch of 3-carbon molecules, and then attached to the CoA molecule and become acetyl-CoA. Now remember, fatty acids or fats have much longer, they have really super long hydrocarbon chains, right? So they have these big long carbon chains. Let's just make this one right here. And they can literally be thousands of carbons long. Now glucose, when it gets broken down in glycolysis, ends as pyruvate. Pyruvate is a 3-carbon molecule. This 3-carbon molecule becomes acetyl-CoA. Well, if I have a fat here that has all of these 3 carbons, 2, 3, 4, I now have 4 acetyl-CoAs from a single fat molecule and only 2 acetyl-CoAs from a whole 10-step glycolysis glucose breakdown. So this is why it's said that fats store more energy than sugars do. But it also takes more energy to break fats down. They still do produce far more ATP per molecule than ATP, but again, they do require more to get things started. After fats are broken down, when you run out of fats, your body will start breaking down proteins. Proteins are energetically unfavorable to break down. It requires a lot of energy to break them down. And if they are broken down, they will enter into multiple pathways. So they enter in all 3 of the different types of pathways. Doesn't mean that we don't break down proteins. A lot of us eat meat, things like that. We do break them down and digest them. But in terms of our own bodies, proteins are one of the last things that our own bodies would try to consume off of ourselves. So that brings us to the idea of a metabolic highway. And the metabolic highway is the idea that cars are kind of like the molecules, the intermediates and precursor metabolites in metabolism. If you're in a car, you can get on the highway, you can get off the highway. You can leave the highway and go become a protein for a while, and then as a protein be broken down and re-enter into the highway somewhere else. So metabolism works a lot like a highway. Aerobic metabolism is not the only form, however. Anaerobic metabolism also occurs. Now, in an environment in which oxygen is not there, we call this an oxygen deficit. There are two different strategies that cells will use. The first is anaerobic cellular respiration. This is most often used, here you have it titled right here. This will use nitrogen, sulfur, carbon can even be used. It'll use something other than oxygen as that electron acceptor. So remember in the electron transport chain at the end of it, when oxygen was accepting those electrons to become a water molecule, when it accepts those, that's in an oxygenic environment. But if we're in an anaerobic environment, oxygen is not available. Our cells can't do that, right? We can't survive without oxygen, but bacteria, some protozoans, there are organisms that are anaerobic and still use cellular respiration. So it's the exact same process. They're just using a different atom at the end of the electron transport chain. Now, because of this, these atoms here do not have the same electronegativity, so their pull of the electrons through the electron transport chain is not as significant, so they tend to produce a little less ATP per glucose molecule than aerobic respiration. Now, if an organism is an aerobic organism, like say our own cells, and it can only use oxygen at the end of the electron transport chain. If it finds itself in an anaerobic environment, some cells can actually carry out glycolysis and use the ATP from glycolysis. In this case, this is oftentimes known as fermentation. So in fermentation, we break down sugar using glycolysis. The pyruvate at the end, instead of being matched with CoA to become acetyl-CoA, the pyruvate is broken down into lactic acid or into alcohol. And during this process, it will cause glycolysis to continue running and produce more ATP. Fermentation produces the least amount of ATP in all of the processes that we've looked at, but it does produce enough for small cells to continue functioning. Our muscle cells, when we are exercising, we may run out of oxygen. We can't get the oxygen to our muscle cells fast enough, and they're using the oxygen and ATP faster than we can produce it. They will then convert to fermentation for a short period of time. That's part of our fight or flight process. So here we have pyruvate is going to combine with NADH to give us an H positive here. Our final product is lactic acid and NAD+. Now down here, we have pyruvate being converted into ethanol and again into NAD+. And you may say to yourself, well, I don't understand. What am I using fermentation for if I'm not getting ATP out of it? Well, let's go back to glycolysis, and I will show you why. Go all the way back here. Okay, in the second stage, right? So this is the second stage of glycolysis, the last 5 steps. This is where we were getting, let me get my eraser out so I can get some of those marks off of there. So I'm going to just erase that and erase that and go back to my pen. Sorry. There should be an easier way to do this. Okay. So in fermentation, I was getting either alcohol or lactic acid, right? Either one of these. and NAD+. And here's why. Every time I want to make an ATP in glycolysis, either here and over here, before either of these ATP production steps by substrate level phosphorylation, in order for either of these two to occur, I must meet this enzyme here and convert this molecule into 3-phosphoglycerate. So this particular molecule here has to be broken down into another one. And in order to do that, it has to be formed. And the formation of that molecule loses an electron and causes us to form NADH. Now, when NADH is formed in glycolysis, it would normally go to the electron transport chain. But I don't have any oxygen. So if I don't have oxygen, my electron transport chain is not going to function. If my electron transport chain doesn't function, I'm not making ATP. So in this case, my NADH has nowhere to drop off its electron. So if I'm going through glycolysis because I want to make these 4 ATP, I want to make these ATP to use them, I'm going to start building up a lot of NADH. And if my NADH does not have anywhere to, if it does not have anywhere to drop off that electron, then all of my NAD plus is going to get used up. How do I combat that? Instead of pyruvate going into acetyl-CoA, which I'm not going to be using because I don't have oxygen, instead my pyruvate gets converted into these two waste products, but it gives me NAD plus. So the whole point of fermentation is to recycle this process right here. As long as I can make NAD+, I can then make NADH. And if I can make NADH, then glycolysis will continue and I can make ATP. So that's the idea behind that. So I'm going to move through these kind of quickly so we can get towards the end here. Now, secondary metabolism is a process where cells are not producing metabolites that are required for cell function. They're usually, they call them secondary because they're not as important, so to speak. They're not required for cells to function. They're not used to produce ATP. These secondary metabolic pathways are used to produce metabolites that are responsible for defense, attraction, protection, and competition. So secondary metabolites work for defense, attraction, protection, and competition. So I'm just going to show you a few examples of those. So the 4 classes or categories of secondary metabolic pathways, the 4 products of those pathways are phenolics. Phenolics are antioxidants that have really strong flavors and smells, usually produced by plants. Most of these secondary metabolites are produced by plants or by insects. Alkaloids are bitter tasting molecules that are used for defense, such as monarch butterflies produce a strong alkaloid that has a really bitter, nasty taste and can even cause sores and a burning sensation in the mouth of birds so that they don't eat them. Now, that alkaloid that they produce has nothing to do with cell structure or growth. It's just simply a defense mechanism. Terpenoids are secondary metabolites usually used for the attraction of pollinators and such in plants, very strong smells and colors. Polyketides are chemical weapons such as toxins that are produced by insects, even some toxins produced by plants to prevent other plants from coming near them. So some examples are, here we have on the left are terpenoids. Beta-carotene is a terpenoid. You may have seen beta-carotene advertised in a lot of foods. It's a strong antioxidant. Beta-carotene is what is absorbed by flamingos when they eat shrimp, and they deposit the beta-carotene in their feathers, and that's what gives them that pinkish color. Here we have phenolics, such as flavonoids and anthocyanins. Flavonoids are found in vanilla. These are vanilla beans. And anthocyanins give a really strong coloration to some fruits and vegetables. Now, why would strawberries want to promote themselves? Well, birds would come along and eat these berries and then poop out the seeds in other areas, and that would allow these seeds to be dispersed by animals. The last two are the polyketides and the alkaloids. The polyketides here, this is streptomycin. Streptomycin is a very common antibiotic. So there's a soil bacteria called Streptomyces that produces this antibiotic. As you can imagine, soil has lots and lots of bacteria in it. So many of our soil-dwelling organisms do produce antibiotic-like compounds in the form of polyketides. They do this to prevent the growth of other microorganisms in their vicinity, so that the limited resources that they have available to them will be available only to the bacteria and not to other organisms that are not part of that species. Then we have atropine, which is found in nightshade. This is a toxin, so that if birds or other mammals eat this particular plant here, they will get quite sick. It's actually a really strong toxin. So that